Hello everyone. This is our first lab video. I'm just going to use this to give you an idea of the equipment that you would be using in the micro lab. Realizing you won't be using it this quarter, but you may be using it in the future. So you should at least have an idea of what's involved. Notice first of all that I've got sort of the standard personal protective equipment on that's common in the microbiology lab. I have my lab coat, I have latex gloves, I have eye protection. We may not always wear these in the micro lab depending on what we're doing, but we've got them in each of the stations. I also have on long pants with closed-toed shoes, as we do in every lab in the biology department. So this would be your station. I'm just going to show you some of the basic equipment that you would be using in a standard microbiology lab. All right. First of all, we have two devices for inoculating cultures, one of which we would use most of the time, the other one would be rare. This is what's called an inoculating loop. If you look closely, you can see that it has it's a wire with a loop at the end, and I'll show you how we sterilize this in a few minutes. We would use this for streaking plates, like I have here, to spread them out and obtain isolated colonies that would be used to generate pure cultures. Okay, so the inoculating loop is used very frequently. This is what's called an inoculating needle. It looks very similar, except that it's just one straight needle. We occasionally use this for stabbing down into media. And I'll show you that probably next week when we talk a little bit about motility. This device is for spreading plates. So it's just basically a thicker metal triangle, as you see here. We would actually dip this into ethanol, catch it on fire to sterilize the surface of it, a little bit different than how we sterilize the inoculating loop. And then we can use this to spread, for example, liquid samples evenly across the surface of a plate, which is quite different than what we do with an inoculating loop. So I'll show you this in a few weeks when we are doing, looking at how you basically count bacterial cultures, count the number of cells in a culture. This is just a flint striker for starting fire. I'll show you that in a second. And then we have things like a labeling marker, a diamond pen for writing on glass, slides, tape. And then in here we have a few other things which I want to show you. This is basically a staining tray. You'll see one of these in the video you're going to watch later on today on the gram stain. This is just an old-fashioned clothespin, which you would use to hold your microscope slide that has a bacterial smear on it while you're doing the staining. A few other things in here that I'm not going to show you right now, but I do want to show you how basically we sterilize the loop. We are going to talk about later in the class today, or the lab today, and on your worksheet, about aseptic technique. So aseptic technique means that we handle all our inoculating instruments in such a way that they are sterilized before we put the specimen on there that we want to, for example, spread on a plate. And then after we're done spreading on a plate, we sterilize it again. So we use a Bunsen burner for sterilizing things like the loops or the plate spreader. This is the Bunsen burner. And so I'm going to put it on the bench top here. You can see it. I'm not going to bother showing you the details of how this works. I would certainly if you were working in the lab, because I want you to be able to use it properly. But since you're not going to be actually doing that yourself in the moment, I won't spend a lot of time on it. This is the gas jet. As you can barely see it on the video. Anyway, we turn the gas on, the gas comes down the burner, and then we light it like that. You can barely see it. It's a nice clean blue flame, and then we use that flame for sterilizing our inoculating loop. So let's see if you can barely see this. You can see that the loop is turning bright orange as it heats up. That kills everything that would be on this loop, dust, dirt, just anything that had settled on the loop. So now when we touch this to, for example, a colony of bacteria, we know that that will be the only thing on the loop, right? I'm going to shut this down now, and then I'm going to switch to another video. So be back in just a minute. You can click on the next video when you're ready.